Welcome to our channel NEETBIO Simplified Biology. In this video, we are gonna see excretory products and their elimination. This video will be helpful to crack NEET and CBSE 11th Biology. In the chapter, Excretory Products and Their Elimination, we are gonna learn about human excretory system, urine formation, functions of the tubules, mechanism of the filtrate concentration, regulation of the kidney function, micturition, role of other organs in excretion, disorders of the excretory system and their treatment. First, we are gonna see what is excretion, their types and their role in body fluids regulation. What is excretion? Excretion is the elimination of both nitrogenous and non-nitrogenous metabolic wastes. It helps to maintain homeostasis of the blood fluid. Nitrogenous wastes include ammonia, urea and uric acids. There are three different types of excretion. They are ammonotelism, ureotelism and Uricotelism. Ammonotelism. In ammonotelism, the organism excrete ammonia through either diffusion or gills. Water required for ammonotelism is more as ammonia is highly toxic, but they are readily soluble. Example: bony fishes, aquatic insects and amphibians. Next, ureotelism. In ureotelism, the organism excrete urea through kidney. Water required is lesser as urea is less toxic than ammonia. Example: mammals, marine fishes, terrestrial amphibians. Uricotelism. In Uricotelism, the organism excrete uric acid in the form of paste or pellet. Water required is least as uric acid is less toxic than ammonia. Example: snails, birds, reptiles. Osmoregulation. Osmoregulation helps to regulate the water and solutes in the body. Types of osmoregulation. First type, osmoconformers. They cannot actively control the concentration of the body fluid with respect to the environment. Example: hayfish, myxine. Next one is osmoregulators. They actively control the concentration of the body fluid with respect to the environment. Example: mammals. Osmoregulation in different environments. In freshwater, body osmolarity will be high and water osmolarity will be less, so water movement will be from external to internal. Ion movement will be from external body fluid to external fluid. Example: fish, amoeba. 2. In marine water, body osmolarity will be less and water osmolarity will be high. Water movement will be from body fluid to external fluid. Ion movement will be from external to internal. Example: marine fishes. 3. In terrestrial environment, different methods of osmoregulations. Example: in camel, they drink 80 liters of water in just 10 minutes and store it in lump. In kangaroo rat, they get 90% of the water from metabolism and store the urea in tissues to extract water. Now we are gonna see about human excretory system. The human excretory system consists of pair of kidney, pair of ureter, one urinary bladder and one urethra. First, kidney. Kidneys are reddish-brown and bean-shaped structures located between the levels of last thoracic and third lumbar vertebra close to the inner dorsal wall of the abdomen. Measurement of the kidney is 10 to 12 centimeters length, 5 to 7 centimeters width, 2 to 3 centimeters thickness, 120 to 170 kilograms weight. Structural and functional unit of the kidney is nephron. The picture shows the pair of kidney from which the ureter arises and leads to urinary bladder and excrete through urethra. There are three types of kidney. First one is pronephric, which consists of only Bowman's capsule. Next, mesonephric, which consists of Bowman's capsule and glomerulus. Example: fishes. Last one is metanephric, which consists of Bowman's capsule, glomerulus and tubules. Example: human. Next part is ureter. It is 20 centimeters long and 3 millimeters thickness. It consists of 3 layers, namely adventitia, outer layer, muscular, middle layer, transitional endothelium, inner layer. These layers are same for urinary bladder. Next one is urinary bladder. As the picture shows, it has a neck region from which the urethra arises. It has 2 sphincters, namely internal urethral sphincter and external urethral sphincter. They play an important role in micturition. We will see what is micturition in the coming slides. Next one is 4. Urethra. It is located in the neck of the urinary bladder. The length of the urethra in male is 20 centimeters and female is 3 to 4 centimeters. That's why female are more prone to infections. Now we are gonna see the detailed structure of kidney. See the picture. 1. Hilum. It is the center inner concave surface through which ureter, blood vessels and nerves enter. 2. Renal pelvis. It is a broad funnel-shaped space inner to the hilum. 3. Calyces. They are the projections of renal pelvis. 4. Outer layer of the kidney is a tough capsule called renal capsule. 5. The inner layer consists of 2 zones, namely, outer cortex and inner medulla. 6. Medullary pyramids. Medulla is divided into few conical masses called medullary pyramids. 7. Columns of Bertini. Calyx, 1.5 centimeters, extends in between medullary pyramids as renal columns is called columns of Bertini. Nephrons. The structural and functional unit of the kidney is nephron. Let's see the nephrons in detail. There are 2 parts of nephrons. They are glomerulus and renal tubule. The picture here shows the glomerulus. Glomerulus is the tuft of capillaries formed by afferent arteriole. Blood from glomerulus is carried away by the efferent arteriole. Next one is renal tubule. It begins with double-walled cup-like structure called Bowman's capsule. Glomerulus along with Bowman's capsule is called Malpighian body or renal corpuscle. Next one is proximal convoluted tubule. This tubule continues with highly coiled network. See the picture here. PCT leads to Henle's loop. It is a hairpin-shaped loop which has both ascending and descending limb. Descending limb continues as highly coiled tubular region called distal convoluted tubule, DCT. Last one is collecting duct is the duct where many DCTs opens. Many nephrons converge and open into renal pelvis through medullary pyramids. Vasa recta, U-shaped. It is a minute vessel of peritubular capillaries which is formed by efferent arteriole runs parallel to Henle's loop. Now we will learn about the location of different parts of nephron in the kidney. Henle's loop is in medulla. Malpighian corpuscle, PCT and DCT are in cortex. There are 2 types of nephrons, namely cortical and juxtamedullary nephrons. Cortical nephrons accounts for 85% of the kidney. Nephrons with 2 short Henle's loop and extends only little into the medulla. No vasa recta and urine concentration. More active. Juxtamedullary nephrons accounts for 15% of the kidney. Nephrons with very long Henle's loop and extends deep into the medulla. Vasa recta and urine concentration are present. Less active. Now we're gonna see about urine formation. Remember the structure of nephron and kidney. Let's see the path of urine. Glomerulus, PCT, Henle's loop, DCT, collecting duct, duct of Bellini, medullary pyramids, calyx pelvis hilum ureter urinary bladder urethra. Now we are gonna learn about the process of urine formation. 3 main processes in the urine formation, namely, glomerular filtration, reabsorption and secretion. 1. Glomerular filtration, filtration of blood in glomerulus. Ultrafiltration is done through 3 layers present in the glomerulus. They are glomerular endothelium, basement layer, between 2 layers, Bowman's epithelium, which contain podocytes which are arranged in certain manner to form a filtration slits. It filter all the plasma without proteins. Glomerular filtration rate is the rate at which the kidney filtrates the blood. It is regulated by the juxtaglomerulus apparatus by producing renin. Total blood flow into the kidney is about 1100 to 1200 milliliters per minute, which is 1/5 of the cardiac output. Glomerular filtration rate is about 125 milliliters per minute or 180 L per day. Next process is selective reabsorption. Total filtrate per day is 180 L and total urine produced per day is 1.5 L. Therefore 99% of the filtrate is reabsorbed by the renal tubules. The picture shows which tubule absorbs which electrolytes. The PCT absorbs glucose, amino acids, Na+, actively, and nitrogenous wastes, through passive. The descending Henle's loop absorbs water and ascending absorbs sodium ions. The DCT absorbs both Na+ ions and water, and collecting duct absorbs most of the water. Then secretion. Renal tubules also secrete H+ ions, K+ ions and ammonia. To maintain the ionic and acid-base balance, both PCT and DCT secrete H+ ions, K+ ions and ammonia. Collecting ducts secrete H+ ions and K+ ions. There is no secretion by Henle's loop. Now let's learn about the function of the tubules. Proximal convoluted tubule, PCT, is lined by simple cuboidal brush border epithelium, which reabsorb 70 to 80% of electrolytes and water. The main function of the PCT is to maintain pH and ionic balance, secrete H+, New Hampshire 3, K+. Next, Henle's loop. Henle's loop. Minimum reabsorption takes place in Henle's loop. It maintains high osmolarity of medullary interstitial fluid. Descending limb is permeable to water and impermeable to electrolytes, so the filtrate moves down. Ascending limb is impermeable to water but permeable to electrolytes, so the filtrate moves up. Next one is DCT. DCT. Reabsorb Na+ ions, water, HCO3, H+ ions, New Hampshire 3, K+ ions. The main function of the DCT is to maintain pH and sodium-potassium balance in blood. Last tubule is collecting duct. It reabsorbs large amount of water to produce concentrated urine. Urea passes through the collecting duct to maintain the osmolarity. The main function of the collecting duct is to maintain pH and ionic balance, secrete H+ and K+ ions. Now let's learn about the mechanism of concentration of the filtrate. This is one of the most important concepts, so listen carefully. Mechanism of concentration of the filtrate is to produce a concentrated urine. Henle's loop and vasa recta play a significant role in this. Countercurrent. The flow of filtrate in the two limbs of Henle's loop is in opposite directions. Same in vasa recta. The countercurrent helps to maintain concentration gradient and increase osmolarity towards the intermedullary interstitium. See this picture. From 300 mosmol per L in the cortex to about 1200 mosmol per L in the inner medulla, sodium chloride and urea. Sodium chloride is transported from the ascending limb of Henle's loop with the descending limb of the vasa recta and returned back by the ascending portion of the vasa recta. Similarly, small amounts of urea enter ascending loop of Henle's loop, which is transported back to the interstitium by the collecting tubule. This is called countercurrent mechanism. The presence of such interstitial gradient helps to an easy passage of water from the collecting tubule, thereby concentrate the filtrate, that is urine, 4 times concentrated than the initial filtrate. Now we are gonna see the next concept, regulation of kidney function. Hypothalamus, JGA and heart plays an important role in the regulation of kidney function. Let's see hypothalamus. Hypothalamus produces antidiuretic hormone, ADH, or vasopressin, which allow the tubules to absorb the water to prevent the diuresis. This process is neurohypophysis. This results in the increased GFR and blood pressure. Next one is juxtaglomerular apparatus or renin-angiotensin mechanism. When the GFR declines, JGA produces renin, which converts the angiotensinogen into angiotensin I and again into angiotensin II. Angiotensin E is a vasoconstrictor which stimulates the production of aldosterone from adrenal cortex, which reabsorb the water and Na+, thereby increases the GFR. Next, heart. The increase in blood flow to heart produces antidiuretic factor, which relaxes blood vessels and decreases the GFR. ANF is a check for renin-angiotensin mechanism. Micturition. The process of releasing urine after receiving voluntary signal from the central nerve system is called micturition. 3 nerves are responsible for micturition. 1. Parasympathetic nerves. Relax involuntary sphincter and contract urethral sphincter. Sympathetic nerves relax urethral sphincter and contract interinvoluntary sphincter. Somatic nerves control external voluntary sphincter. Micturition reflex is the process in which CNS sends signal to the stretch receptors in the internal urethral sphincter, which sends back the signal to the CNS, which in return causes the contraction of internal urethral sphincter and relaxation of external urethral sphincter, and ultimately releases the urine. Urine. pH-OD urine is 6.0 and osmolarity 1.005 to 1.030. Color is yellow, called as urochrome. Smell of the urine is called urinoid. 1 to 1.5 L of urine comes out daily, which consists of about 25 to 30 grams of urea. Composition of the urine includes water, 95%, urea, 2%, electrolytes, 2%, creatinine, 0.3%, ammonia, 0.3%, and hippuric acid, 0.3%. The role of other organs in excretion. Lungs excrete CO2, 200 ml per minute, water, 400 ml per minute. Liver excrete bile pigments, cholesterol, vitamins and drugs. Sweat glands excrete sodium chloride, water, urea, lactic acid, glucose. Sebaceous glands excrete cholesterol, wax, sterols and hydrocarbons. Saliva excrete nitrogenous wastes. Let's see the disorders of the excretory system. Albuminuria, when albumin comes with urine. Glycosuria, glucose with urine. Ketonuria, ketone components with urine. Hemoglobinuria, hemoglobin with urine. Hematuria, red blood cells with urine. Puria, white blood cells with urine. Oliguria, low urine output. Anuria, no urine output. Uremia, increased urea in the blood. Renal calculi, or kidney stones, accumulation of calcium oxalates or crystallized salts in the kidney. Kidney failure, nephritis and the above disorders can cause kidney failure. Glomerulonephritis, inflammation of glomerulus, and it is treated with Streptococcus pneumonia. The treatment of the kidney problems include transplantation and dialysis. 1. Kidney transplantation, isograft, is the transplantation of tissues between identical twins. Allograft is the transplantation of tissues of different individuals. 2. Dialysis, hemodialysis, artificial kidney is used outside the body for dialysis. The unit consists of cellophane tube and surrounded by dialysis fluid, which consists of the same composition as plasma except nitrogenous wastes. The unit is added with a heparin, an anticoagulant agent, before the blood passes through the artificial kidney. The cellophane tube consists of small pores through which the blood filtrate. The anti-heparin is added after the filtration and passes through the veins to the body. Peritoneal dialysis occurs inside the body in the peritoneum of the abdomen. The picture here shows the hemodialysis, where the heparin is pumped into the artery where the blood passes through. After it is dialyzed through the artificial kidney, anti-heparin is added and passed into the body through veins, and it continues. Excretory organs in different organisms. In most of the invertebrates, excretory organ is kidney. In Platyhelminthes, rotifers, annelids and cephalochordate, it is protonephridia or flame cells. In earthworm, annelids, it is nephridia. In cockroaches, it is Malpighian tubules, and crustaceans like prawns have antennal or green glands. We saw the excretion chapter in detail. If you have any doubts, please comment this video. Thank you for watching. Please do like, comment and share. Subscribe the channel more videos and click the bell icon to stay tuned.